students. Today we're going to very quickly finish our notes and move right into our lab activity for this week. So as we finish up our notes, let's remember our theme of this week. The topic of the week has been stewardship. And we're going to be finishing up today with specifically animals and even more specifically, the whooping crane. But in more general terms right now, biologists, wildlife biologists in particular, like to keep track of animals. We've seen a couple videos earlier this week already where organisms like the bald eagle and the California condor have been kept track of. Well, there is something called tracking of animals. And what we do is use a technology called telemetry. Telemetry is what scientists use to keep track of animals using an electronic beacon. And this beacon is a device that is placed on the animal after it is trapped or captured and then released with this beacon on it. And the beacon releases a radio frequency. And this radio frequency can be tuned into using telemetry. And so this telemetry uses antenna like this hooked up to devices that can actually pinpoint the location of anything. And in our case, can identify the location of animals or whatever else it might be that is being tracked. So let's move into our lab activity. But before you turn the page, what we're going to do first, on the bottom of your notes page, you'll see some pictures there, including one of the whooping crane and a graph there. And underneath there is some space to record some notes regarding a video, because the first thing you're going to be doing is watching a little information about the whooping crane in a video. And I'll link you to that right after I finish this video. So you're going to watch that video first. After watching that video, then you're going to start the lab activity, and you'll have to turn back in your books to page 207 for that particular activity. And the problem, I'd like you to read and write a hypothesis when it comes to that time. And the problem is, in what ways does weather affect the reproduction rate of whooping cranes? And essentially what you're going to be using is historical data, actual data from the years 1955 to 1970. And I know this graph is a little bit small for you to actually see, but the years are on the left, 1955 all the way down to 1970. And there are columns here with data that you're going to be using to complete your lab activity. The first column is labeled migrating adults. And I'd like you to write something at the top of that column, if you would, please. I'd like you to write population, because later in the lab activity, when you start graphing, you're going to have to graph population, and it is actually that number that you're going to use to graph the population. So that first column, at the top of that first column, label it population, above where it says migrating adults. Now, I've given you instructions in your lab activity of how you are going to calculate this open column entitled hatching success. And the hatching success, according to the procedures, are basically is this. This is how you can calculate it. It is the number of eggs hatched divided by the number of eggs laid for a particular year, multiplied by 100. And I've given you that equation there, and you can use a calculator to help you, but you'll need to fill in this column in order to use that data on your graphs. And I just want to make mention here very quickly of the two different graphs you're going to have to make with this activity. The first one is simply a population graph. Along the x-axis, each one of these lines represent 1 year. So the first line is 1955, and the last line is 1970. On the y-axis, you're going to label the population in terms of the numbers from 0 to 50. And so the bottom line will be 0, top line will be 50. You find the correct interval to label the y-axis appropriately. But now I want to mention the second graph here before we get to our video in a moment. The second graph is going to be a double line graph, and I'm asking you in the procedures to use two different colors. But if you don't have two different colors at your disposal, then make one line a solid line, make one line a dotted line if you wish. Two colors would be great, but if you can't do two colors, then two different kinds of lines would be fine. And this one is a really important graph because you're going to be using this graph to make some conclusions. The x-axis, again, is the same as the first one, the year from 1955 to 1970. So that's pretty straightforward. Now, the y-axis, you actually have two different y-axes. The y-axis on the left, you're going to label from 0 to 100. 0 at the bottom, 100 at the top, and this is the hatching success. This is the column that you're going to be calculating during the lab activity. On the other y-axis is the precipitation from 0 to 10. So label this axis appropriately as well. And this is where your two different lines come into play. One color will be plotted using the y-axis on the left. The other color, or the other kind of line, will be used using, or will be made using the y-axis on the left. Both of them use the same x-axis, however. So you're going to have two lines on this graph, one for the hatching success year by year, one for the precipitation year by year. And what you will need to do in order to make your conclusion is look at the relationship, if any, between the two lines on this graph in particular. So take your time, calculate your data, graph your data, be as neat as possible so that you can use your graphs to make your conclusion, which will answer the problem at the top of your lab activity. So I've talked enough. I want time to give you to watch the video on the whooping crane first before you begin working on the lab activity. So for now, I'm just going to say bye-bye.